Well, minimal residual disease negativity is a clear measure of the effectiveness of a treatment, and it's an evaluation that can be performed quite early in the course of the disease compared with other endpoints used for drug approval, such as progression-free survival. So it has the advantage of providing information early on, and so potentially speed up the process of drug approval and also drug development, and consequently, in particular, the drug availability for patients. MRD is prognostic, so achieving minimal residual disease negativity deeply correlates with the progression-free survival as well as with the overall survival. And this has been seen in several settings: newly diagnosed patients, relapsed patients, high-risk disease, as well as standard risk disease. So this is one of the most important endpoints for cancer treatment in terms of efficacy of therapy. First of all, MRD needs to be standardized in terms of which test should we perform in which patients, when we should also add to the bone marrow evaluation the imaging test. We have to define if it's MRD per se important, so the achievement of a single evaluation that is negative, or the durability of the minimal residual disease negativity, which is likely to be even more important than simply achieving one single MRD negative evaluation. Then we have also to understand in which time points we should evaluate it in the setting of both a sequential therapy as well as a continuous treatment approach. So there are many issues that are under investigation in an effort to standardize the procedure so that you can define how to use it in clinical trial, because at the moment it's only used in clinical trial, and then once you have shown in clinical trials what you should do, then you can translate it to clinical practice, hopefully.